Welcome to Microbiomes Lesson 2.5, Analyzing Evidence About Fecal Transplants. Ooh, it's going to get good. Fecal transplant evidence. I can't wait to find out more. It's so intriguing. First thing, as always, is our warm-up. Review the new data about patient 23, then answer the question below. Well, let's check that out on the Amplify website. It looks like this. Looks like a dish. Oh, here we go. Then answer the question. So here's our information. Oh, look, week 7 and week 9. We haven't seen week 9 yet. And what happens in this period? The patient gets a fecal transplant. And look, in case you were wondering, they colored it brown, just so you would know what's actually in that little vial. So what differences do you notice in patient 23's gut microbiome between week 7 and 9? Record at least 2 observations. I want you to look at this, and then I want you to look at this, and give me 2 observations. Go ahead, take a minute, do that right now. On your marks, go. How'd you do? I'll tell you what my observations were. Number 1, I noticed, well, this red thing isn't there anymore. What was that red thing? If we zoom in, oh, C. difficile bacteria. I see. Also, another thing I noticed, well, shoot, look at all this empty space. It got filled in. Got filled back in. Look, this little one here, bacilli, that one, it's way big now. So it looks like this bacteria, this helpful bacteria, has taken over again, and it booted out the C. difficile bacteria. Imagine that. That's just fantastic. So let's keep moving on. Discussing new information from the Institute. We're going to start off with a key concept, actually, number 10. This is our last key concept for this unit. Living things with fewer than normal helpful bacteria in their guts can become infected more easily because there is more food and space available for harmful bacteria. Makes sense, right? Hey, look, we got a bunch of food over here. Oh, cool. Well, I'm going to go over there. Hey, we got a bunch of space over here. You guys all crowded over there? Oh, yeah, shoot, that looks better over there. So there's more food, there's more space. That means, yeah, other things can move on in and survive and live. Did we notice that? We sure did. Look at all this space and food. And then once I got the fecal transplant, it filled on in. Hey, remember Robert, Bobby Naismith? Remember the senators claiming that tiny microorganisms are too small to affect human health. I mean, come on. We learned about this like so long ago. Based on this claim, he wants to cut funding for fecal transplant research. Well, I mean, I can see cutting funding for fecal transplant research for other reasons, but not based on the fact that microorganisms are too small to affect human health. Come on. Hey, look, we got a letter. We got new information from the Institute. It's kind of hard to read right here, so I'm going to pull it up on our Amplify website. Ooh, look, it's from Dr. Mara, 2 student researchers. That's us from the Microbiome Research Institute. From Mara, head scientist, subject, fecal transplant procedure presentation. Attachment. Ooh, there's an attachment? Crazy. FT procedure. I'll bet that stands for fecal transplant procedure. Thank you for your careful work analyzing the data for patient 23. As you know from the politician's speech, the fecal transplant procedure has something to do with feces, poop. I've sent you a slideshow presentation to review that includes a detailed explanation of how the procedure works. Don't know that I want to view that presentation, but okay. I think this information will help you understand more about how fecal transplant helps cure a patient infected with harmful bacteria. We also want to include your research in a press release in which we will publicly present our arguments against the senator's effort to cut our funding. They want my research? In this press release, we are going to present our evidence in support of this claim. A fecal transplant can work to cure a patient infected with very harmful bacteria such as C. difficile in many ways. Okay? We think we have strong evidence to support this claim, but we will need your help to research and write a convincing scientific argument. Well, Dr. Mara, I think we're up to the task. Let's take a look, a gander at this presentation, this fecal transplant presentation. Here we go. Notes about, a fecal transplant procedure. Feces contain the microorganisms that live in a person's gut microbiome. Okay, so yeah, the microorganisms don't just live in your gut, but they actually make their way into your feces or your poop. So Poop is full, chock full of microorganisms. All right, again, there's our nice little, thanks for coloring it brown, MRI, we didn't realize. Notes about a fecal transplant. Okay, here we go. A fecal transplant is a treatment where a doctor takes the feces from a person with a healthy gut, this person right here, and puts it into the gut of a person with an unhealthy gut microbiome during a medical procedure. Okay, so we take the feces and we put them in this thing, and then we put them in this person. Great. After the transplant, the microorganisms living in the feces of the healthy person now live in the gut of the unhealthy person. Okay, so you're just transplanting the microorganisms from one person to another person. Okay, that makes sense. In their new environment, microorganisms use the food and space available in the gut to grow and reproduce. In just a few days, the total number of microorganisms, including helpful bacteria added to the gut microbiome of the unhealthy person, is 75 to 80 trillion. That's crazy. Just a few days and we reach those numbers. That's nuts. These bacteria, these microorganisms, they reproduce crazy fast. All right. Let's keep going. Adding more helpful bacteria, microorganisms into the gut is one way the fecal transplant procedure helps secure a patient with a very harmful infection like C. difficile. When more helpful bacteria take up space and food in the gut, there is less space and food available for harmful bacteria. So we're adding a whole bunch of good bacteria so that the harmful bacteria is like, oh, wait, hey, no, I wanted some food too. Oh, no, I wanted some space too. Oh, no. And it's like, no, get out of here, harmful bacteria. I get it. All right. Okay, I guess that was the end of the presentation. What unanswered questions do you have about the fecal transplant procedure? I have a question. I have a question. So how do they do it? We're not going to talk about that because honestly, it doesn't give us that information. It is a medical procedure, though. But I know. I mean, if you're curious like me, you want to know more about it. All right, so there it is, putting the healthy feces from a healthy person's gut into the gut of someone who is unhealthy. All right, here we go. We have some claims and subclaims. Here's our main claim. A fecal transplant can work to cure a patient infected with very harmful bacteria such as C. difficile in many ways. There's many ways. Number one, bacteria from the fecal transplant can fill up the space in the gut, which limits the food and space for invading harmful bacteria. Cool. Yeah, we've talked about that. This new bacteria fills in all the gaps, and the harmful bacteria have nowhere to live. Subclaim number two, bacteria from the fecal transplant can help the patient's body produce immune cells that kill invading bacteria. Well, that's interesting. Didn't realize that these bacteria were so dang helpful. Another one, bacteria from the fecal transplant can help the patient's body produce mucus that protects the gut from invading bacteria. Jeez, all this helpful stuff from the fecal transplant, these microorganisms, they're doing so much. Producing mucus to protect us, helping us to produce immune cells, and taking up all the food and space that this harmful bacteria wanted. Well, we're only going to focus on one of these subclaims, and that's the first one, the one that we've been talking about. The bacteria from the fecal transplant can fill up the space in the gut, which limits the food and space for invading harmful bacteria. So what are we going to do? We're going to create an argument. All right, so we are now in activity 3. So in activity 3, we're going to start talking about this thing called reasoning. So we've got a claim, right? We've got a bunch of evidence, but sometimes the tricky thing is talking about how does that evidence actually support your claim. So let's talk about that right now. First, let's start with this example. Here's our argument right here. Drinking cola is bad for your health. That sounds like a claim. Cola contains 4-MeL. That sounds like evidence to me. All right, so is this argument convincing? Why or why not? First of all, go ahead and just pause right here and think about that. Is this argument convincing? This argument has a claim, like we identified. It has some evidence, but what is missing here? Well, let's take a look. Remember, part of creating a strong argument is making your reasoning clear by showing how the evidence connects to the claim. Okay, so the evidence is that it contains 4-MeL. The claim is that cola is bad for you, but we're missing that reasoning part, aren't we? One thing we're going to be doing is we're going to be using this reasoning tool. We're going to use this all year long for all of our arguments. and it's going to help us. So we can put all of our evidence on this side. Hey, our evidence is that cola contains 4-MeL. And we'll put our therefore right here in this column. We're going to put our claim. Cola is bad for you. But why does this evidence matter to this claim? Let's keep going with our cola example. Oh, there we go. Why does this evidence matter? That's the part that's missing. Here's our evidence. Here's our claim. But we're missing this box. Again, this can be the hardest part for a lot of people. Oh, check this out. 4-MeL is a food coloring that has caused cancer in lab rats. Oh, dear. Many things that are bad for rats are bad for other living things like humans. Okay, well, now I understand why saying cola contains 4-MeL might lead you to think that drinking cola is bad for your health, because you explained what 4-MeL is and what effect it might have on humans. So, lost it. If we add that into our initial argument, here's what it would sound like. Drinking cola is bad for your health. Cola contains 4-MeL, which is a food coloring that has caused cancer in lab rats. Many things that are bad for rats are bad for humans, or for other living things such as humans. Therefore, drinking cola is harmful to the health of humans. Well, I can think of other reasons why drinking cola is harmful to the health of humans, but apparently this is one of them, according to this argument. So we can see how this argument is way stronger than our initial argument, because we're saying why. Why does that matter? So for the rest of our lesson, we're going to be working with this subclaim, just like we said. Okay, there's going to be some pieces of evidence. We have three pieces of evidence, and we're going to discuss each one of those when we come back to class. You're going to think about it on your own first. You're going to write some things about it, but when we meet together again, we'll discuss it too. So the first piece of evidence right here is that patient 23's health before and after the fecal transplant. It's a little hard to see on the slide, but you have these in your Amplify. Boom, there it is right there. Here's the procedure we're supposed to go with. Partner A, you don't have partner A or partner B. If you can, hey, contact somebody in the class, and great, good job. Otherwise, we'll discuss this when we meet as a class too. But on your own, you can also do this. You're going to describe the evidence. Hey, what do you know? It looks like we have an empty gut. Not empty, but a very less than full gut here with some C. jejuni bacteria. Then the patient was experiencing these symptoms. We gave the patient this fecal transplant, and now look, the C. difficile bacteria looks like it's gone. It looks like all the other healthful bacteria have repopulated and filled in the gaps there. Okay, great. So what are we supposed to do? What are we supposed to actually submit here? Here we go. Apparently nothing. But you have three of these. You have three of these pieces of evidence that I want you to think about. And I want you to really try to figure out, why does this matter? Why does this support our claim? You don't have to write anything here, but you will have to write anything. You will have to write something next. So we have that one, the mice experiment with the salmonella. Remember that? We have this article, the human microbiome. We're going to use all three of those. Okay. So let's go ahead and jump to the reasoning tool. First, go through those pieces of evidence. Make sure you've done that. Make sure you can explain what each one is. Make sure you can explain why it's helpful to supporting our claim. Please go and do that right now. Next, we are going to now use this reasoning tool, which again, we're going to be using this all year long for every single one of our units. There we go. We will use the reasoning tool to prepare an argument about patient 23. It will help us explain how each piece of evidence supports subclaim 1. We'll review how to use the tool and then fill in the first row together. So in the first column, hey, look at that, in Amplify. I'm in Amplify right now. Oh, it already filled out all the evidence for you. Each one of those things. Okay, well, that's fantastic. Thank you. In future lessons, you might have to go find your own evidence to put into the reasoning tool, okay? In the middle column, we'll explain how the evidence supports the claim, and then we will write all or part of the subclaim in the column on the right. So we can actually fill out the right column right now, because what is the right column? The right column is fecal transplants can help cure patients infected with harmful bacteria, right? Isn't that our claim? Because they take up space and food that the harmful bacteria might use. It was something like that, right? Well, you'll write that in the therefore column. So what you need to do now, though, is you need to really focus on that reasoning column. Why? Why did patient 23's case study data for weeks 7 and 9, why does that support our claim? What about our salmonella bacteria experiment with the mice? Why does that support the claim? Because you wouldn't just say, well, because experiment 1 of salmonella bacteria, because patient 23's case study. Well, what about it? What about it? Tell me why. Why does it matter? Okay, and again, that can be really hard, especially for somebody who's never done something like that before, never been asked to do that before. So go ahead and work on that right now, hit pause, and come back to me. Last thing is the homework. It's all homework. Well, you're going to finally write a little paragraph for this press release, right? Remember the one that Dr. Mara said that we were going to be helping with? So here's what you do. You're going to use your reasoning tool. If you filled this out already, most of your job is already done. It's just putting all of this into paragraph form. You have so much of the wording already. Okay, so let's go ahead and jump into number 5 here. Let's see what it looks like in Amplify. Ooh, press release, MRI. So, oh, nice. They give us some sentence starters here in case you're having trouble figuring out how to word maybe your paragraph. But here is our subclaim right here. Bacteria from the fecal transplant can fill up the space in the gut, limiting the food and space for invading harmful bacteria. Write a paragraph that supports subclaim 1, include evidence, we have that, and explain how your evidence supports this subclaim. That is the reasoning. So use the reasoning tool. Refer to the scientific argumentation sentence starters, right there. And lastly, the Microbiome Research Institute will publish this press release as a way to inform the public and defend their funding. So that's why you're writing this. You're writing this to say, hey, check out our evidence. Let's not stop funding fecal transplant research. Let's actually, continue it because it might actually be helpful for some of these people who are experiencing these problems. So that's the last part of it. Write that paragraph. I can't wait to see what you come up with and give you some feedback on those. Good luck. See you next time.